All right, good morning, good afternoon, good evening, depending on what time it is. Well, this is our sixth chapter 11 video notes for A Twisted Tale of Hard Work. And of course, we're talking, as we've been studying with these mysteries, we're learning about DNA and how it makes the body protein. So our DNA is working all the time to build our proteins. We're going to study a little more today about problems 1 and 2, but we're going to bring problems 1 and 2 kind of to a close. By the end of these slides, and after all the stuff we've done, hopefully we have a good sense of what DNA is doing, how it's keeping us healthy, and we should have a sense of what some proteins do for us, and why we want our DNA to make the protein. Hopefully today also in our sixth video, we'll start addressing problems 3 and 4, which is, what do we call it when something happens to our DNA? What kind of lifestyles do we choose that can affect our DNA? So we'll start exploring what a mutation is and why we should care, and how it poses risks to us. All right, join me as we go. All right, we last left off on ribosomes, and from a lot of simulations we did, we found out that ribosomes really coordinate these different activities to help make a protein. Let's go back to a quick simulation and check it out. Here we go. Well, the ribosomes, which are the big brown thing, they allow these tRNAs to come in, these green guys. And as the tRNAs come in, they bond these smaller molecules with 3-letter names. These little circles are amino acids, and as these amino acids form bonds together, they start forming the beginning stages of a protein. It's going to go and go and go, and eventually it's going to make a fully formed protein. Now, in the simulation, it really doesn't. In the ribosome, you can see it takes off. This right here, we don't know what its function is. We just know that proteins have a lot of important jobs in the body. This protein is built of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 amino acids. So amino acids overall form the protein, and proteins themselves have a repeating amino acid structure. That means they in fact are polymers. So proteins are polymers, because just like DNA, they have a repeating structure. Now, just a quick review, like we've been talking about, is proteins do a lot of important jobs. We have a protein like we just saw here. Well, maybe it's going to be a special protein fiber in the muscle cell, so you can generate force and power. Maybe this protein is going to become and serve an enzyme function, where it helps chemical reactions occur like digestion. Maybe this protein is going to go to become part of a channel or pump, the way the cell decides what goes in and out of it, to keep you alive, keep the cells running healthy. Perhaps this protein will function as a hormone that is determining how well you sleep at night, or how you feel emotionally, how your body develops, how your cells develop as you get older. And maybe it's this antibodies. Now, antibodies are special proteins I need right now, because believe it or not, I'm sick, and I probably have viruses in my blood giving me the symptoms of a cold, right? Runny nose, sneezing. Well, a protein like this can end up in my blood and help identify the viruses, label them for removal, protect me. Don't get sick. Alright, slide 36. We got a ribosome here, right? Well, turns out there's a special way that these tRNAs come in. So this is our mRNA, right? Well, tRNAs connect to the mRNA in a special way. Every three nucleotides of mRNA code for a specific amino acid. That means AUG is a code for MET. Check this out now. The next code is CCA. So this tRNA will only connect to code that's CCA, and it brings in a specific amino acid called PRO. Now, the next section I can't see because the ribosome's in the way, the outside ribosome, but the next code is gonna end in GC. So every 3 nucleotides are a code for a specific amino acid. Okay, so it's GGC. GGC is a code for glycine. It's an amino acid called glycine, and this tRNA brought in the glycine. So every 3 nucleotides codes for a specific amino acid. GGC. What was GGC again? So it's another glycine. Let's look at the definition. Let's get that. So in your notebook, go ahead and pause this video just so you can get the definition. It says 3 nucleotides equal 1 amino acid equals building molecule for a protein. So every amino acid helps build a protein, and every 3 nucleotides is code for 1 amino acid. There's a word for this, and we're going to study it. It's called codon. So every 3 nucleotides is called a codon, and it's called a codon because it codes for 1 amino acid. Alright, the rest of this slide is studying this model here. Let's make it a little bigger. Alright, well, it says what 3 mRNA nucleotides codes for the MET amino acid. So we got the MET amino acid right here, and you can see, just like in our simulation earlier, it's AUG. So the 3 mRNA nucleotides that only code for MET, or the MET amino acid, is AUG. Let's check this out now. It says what 3 mRNA nucleotides code for the PHE. And so you'll scan until you see the PHE amino acid here. So it's the gray circles. Now we already know MET is AUG, and you find PHE here, and it's UUC. So now you have the answer to both questions here on slide 36. Make sure, of course, you've labeled slide 36 in your notebook. Alright, slide 37 here. Let's check this out. It says, interpret and apply the amino acid code to produce a small protein below. It says, draw connect amino acids chemically bonded. So in your notebook, I'd like you to create 1, 2, 3, 4 circles. So put 4 circles and connect the 4 circles with a line, and the line will represent chemical bond, chemical bond, chemical bond. So we want to chemically bond 4 amino acids. Now, inside the circle here, don't write the letters. These are the mRNA codon right here. But GUG, what we want to do is, we don't want to write GUG in our notebook. I want you to write the amino acid. And the amino acid are these little circles here, like LEU. LEU is the amino acid. Let me show you what I mean. Okay, so GUG, that's the mRNA, right? So GUG is right here, and GUG and any other 3 nucleotides here codes for one specific amino acid called val, or valine. So what I want you to do is write valine in the circle. You got these 4 circles that you've written in your notebook for slide 37, and you've connected them with a chemical bond. But of course, I want you to take the RNA code here and replace it with a protein. So in your notebook, you'd write val, VAL, for the val amino acid. Sorry, it's so blurry. All right. Now that you get the picture of what I want you to write in, because this is the amino acid, now you should do CAA. And you look at the code and you look for CAA. See if you can find it. CAA, CAA, CAA. Did you find it? It's right here. GLN. So GLN, this gray circle, is the next amino acid. I'd like you to draw that in here as a circle and write GLN for the next amino acid. And then you have UUU right here. It's the PHE amino acid. So the nucleotides UUU and RNA code for the PHE. So you grab this little circle and draw it into this bubble here in your notebook. And then lastly, you have UAA. And UAA means stop. So you'd write the word stop inside this bubble. It's not so UAA doesn't actually code for amino acid. It's a code for, so anytime RNA has UAA, it means stop the production of the protein. You're good to go. All right. Now, once our DNA has coded for all these amino acids and bonded them together, you have the protein. Once that protein is formed, it floats around. As the tRNAs come in and they link these amino acids all together, every 3 RNAs code for a specific amino acid, and they go and go and go. Every 3 RNA codes for 1 amino acid. And you can see each of the amino acids are modeled as circles, and they all have a 3-letter name. Now, once this happens and you have bonded all the amino acids together, it's called translation, because we've translated nucleotide code to protein. So we've translated the code of RNA and DNA to a protein. Translate. Let's look at Here's slide 38. Make sure you write slide 38 in your notebook. And let's copy down the definition for translation. So the official name for ribosomes, making DNA code turned to amino acids and finally a protein. This whole process here is referred to as translation, right? And it's the process of converting an RNA into a sequence of amino acids that make up a protein. Alright, next it says, how is translating a language like DNA translation? Why do you think they called it translation? How is taking DNA to amino acids to protein the same as like translating one word from one language to another? How do you think that is? Go ahead and write your answer to that question in your notebook. And then next thing it says, organize in correct order the process of chemical codes. Which one of these, so you put A, B, and C in the right order for making a protein. Which is first, DNA, RNA, or amino acid? Which is second? Put them in the order that you believe is correct for making proteins in the body. And then lastly it says, interpret this image. So this is our image right here. DNA, RNA, amino acid, protein. It says, how does amino acid chain form a protein? So look at this one right here. So you see a straight chain of amino acids. And then in the next picture, you see it doing something. So how does this become this shape here, this three-dimensional structure? This is a one-dimensional structure. This is a three-dimensional protein. So how does a one-dimensional chain or amino acids bond together form a three-dimensional protein? Write it in your own words just by looking at this picture and kind of use this as a little helper of how this goes from this to this three-dimensional structure. Give your best thinking on it. Alright, lastly we have slide 40, and 40 is an activity we'll do in class. We're once again looking at the pleasure receptor gene, the gene that makes this little protein here, the little Y-shaped thing that bonds to the emotion chemicals in certain parts of the brain. Well, this time though, you're taking the original DNA of the gene, and since we already know the RNA code of it, you're going to take that RNA code and decide, well, what amino acids does it make that will actually contribute to the overall protein structure? And we'll use a little code like this, but just remember, proteins basically are made simply of these amino acid molecules. And they bond together and they'll form this really impressive, huge protein polymer that it takes a whole, folds and takes this whole like three-dimensional structure. But in class, you'll be filling this out. You'll work with your team. And if you miss this day, just let me know and I'll hook you up with the worksheet so I can get it done and learn a little more along the way. But basically, it's all about translation. How a protein finally gets turned from DNA to a new thing called that protein we've been learning so much about. Alright, good luck and have a great day.